Hello everyone, thank you very much for being here and for hearing me give this talk. So I'm going to be talking about whether they're actually safe to eat and what the current requirements are for safety assessment and how inadequate they are and what actually happens if you exceed them. So some of my background's been given. I'll give a little bit more here. I've actually got an honours degree in organic chemistry, so I used to make chemicals. I've got a PhD in medicine in the field of nutritional biochemistry, metabolic regulation and lots of animal feeding studies. And I've also got a Master of Public Health, which makes me an epidemiologist and biostatistician. I taught at an agricultural college. I've worked in the fields of HIV/AIDS and nutrition, as well as in infectious disease control, investigation and control. I was the senior epidemiologist in my state government's communicable disease control branch, as well as being a member of ARTEC of the United Nations. So we'll get on to this now. So I'm going to start by just saying, well, how actually are GM crops made? Because it all boils down really to that. One of the traditional ways of making it, and these are foods that are still in the food supply, is to use the biolistics method. So this is a means of actually basically getting bits of DNA that you hope that the plant might take up, and using a modified shotgun to try and shoot those bits of DNA into a plant and hope that the plant will take up those pieces of DNA and actually then express them properly without any faults being made. One of the problems with that is that it's actually inserted randomly and may actually affect the function of the plant. There might be new substances produced. The bits of DNA that go into the plant include bits of DNA from plants, animals, bacteria, fungi, viruses, etc. And there's also antibiotic resistance is often put in there, and I've seen one GM plant that was a potato actually that was made resistant to 5 antibiotics at the same time. So why do this? Most plants are genetically modified either to be resistant to a herbicide. So this means that now you've got your field of, say, soy, you can grow your plant, you can then spray it with a particular herbicide, and the plant you want lives while the weeds around it die. Now the main one here is actually glyphosate, which is the active ingredient of Roundup and various other herbicides, but the main one is Roundup. Another type is designed to make its own pesticide, and already we've heard in the previous talk a very excellent, giving you an excellent understanding, I hope, of these insecticidal proteins, which are called the Bt proteins. Now the thing about these is that they're made in every cell of the plant. So if you actually spray a crop with Bt, the organism, the bacteria, you can then wash that off afterwards. If the rain already hasn't done it for you, you can wash it out under the kitchen sink. But you can't if the plant itself is making it in every cell of the plant. You can't, there's no withholding period after the spray because it's in the plant and it's made in every cell of the plant, and you can't actually wash it off. Now, quite often these days we're actually getting both of these being put into plants at the same time, so you're getting what's called stacking of genes. And a triple stack is very common now in the United States, where both of these things are being put into the plant at the same time. And one 8 stack has now appeared, so that's 8 GM genes being put into the plant expressed at the same time. So let's have a look to see what the gold standard is of safety testing for anything, and this is particularly occurring with pharmaceutical drugs. So if there's a new aspirin, for example, comes onto the market, this is what needs to be done. First of all, you undergo thorough animal testing before you go anywhere near a human being, thorough animal testing to determine what effects, good and bad, it has on the animal. If it passes that test, you then take a small number of healthy volunteers and see if it causes problems. If it passes that small number of volunteers, does the thing actually work the way you want it to? If it passes that, you get a lot of people, you randomise them into having the new drug, not having the new drug, and you actually see what the consequences are. And this is all double-blinded, people don't know what they're on, etc. Then if it passes that, it's generally monitored in the environment, monitored in the community to see if something else might be coming out in health effects. And then there's a new step, which is actually getting a lot of randomised controlled trials together and looking at the overall effect that you can see from all of these trials when you compare them. So how far through this gold standard have GM crops come? Well, let's have a look, shall we? Let's see what sort of safety assessments regulators need. Now, this is Food Standards Australia and New Zealand, and that's the food regulator for Australia and New Zealand combined. It's 2 countries at the same time. and it regards itself as one of the best in the world. So I actually looked at 12 reports that Fazance did. So this is a GM company, GM crop company, comes to Fazance, says, we've made a new GM crop. Here is our safety information. Would you like to please assess it to be safe to eat? And I looked at the reports that Fazance actually wrote on these things, covering 28 GM crops, to try and work out, well, what does a regulator need, what's given to it, and how does that compare to the actual gold standard? There are four main things I think that need to be covered. One is, what's the allergic effect? Does it cause an allergy? Does it cause cancer? Are there reproductive problems? And does it cause a toxic effect on, say, your livers or your kidneys? The allergies are actually done on paper only. They're not actually fed to animals or to people. There are some blood samples that sometimes might be tested, but that's not. But we're talking about a new crop here expressing new proteins which haven't been seen before in the human food supply, so you would not be expecting to find an allergic response. You actually need to be exposed to it first and to build up that exposure, generally speaking, for an allergy to develop and then to get worse. They're not fed, animals aren't fed long enough for cancer to develop and so forth. So what does Fazance actually require? Well, really all it requires is a compositional comparison or substantial equivalence. Now this is fairly standard for regulatory bodies around the world, and it was also standard for, my understanding is, the European Food Safety Authority, although now that is requiring extra work to be done that my food authority still does not require to be done. So my food authority says, all right, we're going to see if there might be a new protein being produced. Now, but it doesn't look at the new proteins so much as it actually accepts from the company making the crop that those proteins will be broken down into the building blocks of the proteins, which are the amino acids. It usually doesn't even look at the components of fats, sometimes maybe trypsin inhibitor and so forth as anti-nutrients. Now, if the problem lies with potential proteins that the crop might be producing, what happens when you break those proteins down and look at the amino acids? Well, it's a bit like comparing the fire safety of this building to the one next door, not by looking at where the fire stairs are or the fire extinguishers, but by bringing a bulldozer in, knocking the two buildings down and counting the bricks. You've just lost all of the information you need. So the other thing is that there are some very small sample sizes being used, often 5 to 7, as little as 1. Now you get what's called a type 2 error being produced in this, and that is you may have something screamingly different between a GM crop and a non-GM crop, but if you've got very small sample sizes, it won't reach statistical significance. If it doesn't reach statistical significance, it does not exist. So also the statistical information that was given is not the sort, it wasn't anywhere near adequate enough for a peer-reviewed scientific journal. There's no definition of what constitutes substantial equivalence. What's the pass? What's the fail? What level, when you go below or above, do you say it's not substantially equivalent? If there's no definition, everything passes. If there's no speed limit, no one gets booked for speeding. And there was one corn variety where almost half of the amino acids were statistically significantly different, even on small sample sizes. And my food authority still said it was substantially equivalent and could come into the food supply. It does not require any animal studies whatsoever, no human studies whatsoever, and it takes whatever data the industry gives to it and the explanation the industry puts on the data and accepts that. It does not go into the raw data to check to see whether the industry has said what's in the data is correct. There's that lack of due diligence as well. So it relies upon the industry interpretation of the data. Now, worldwide, there's even less required for crops with stacked genes. So basically, if a gene has been accepted to come into, say, maize, that gene into maize, that gene into maize, that gene into maize, you can put them all together into maize and you don't need any further testing at all because they've all been found to be okay. So there's no measure of whether there might be any synergy occurring between those genes, any additive effects or any synergy. It's all assumed that that won't happen. So as I mentioned that there was some toxicity studies that were done. Let's look at the standard toxicity study now. So this is done by the industry. And this is an acute toxicology study. And now this was all I found being done on 61% of those crops that I looked at that Fessant said were safe. Basically what they do is they say, we've genetically engineered this crop to produce that protein. We're now going to get that protein only, and we're going to put it down the throats of some rats, and we're going to see if they die within 7 to 14 days. And that, if they don't die, everything's fine. But statistically significantly more have to die in the GM-fed group than the non-GM fed group, okay? If some die in the GM-fed, but it's not statistically significant, everything's fine. So what are the assumptions here? Well, first of all, that crop will produce the GM protein and nothing else will happen. No other proteins will produce, no other substances. Also that the protein that they actually put down the throats of the rats doesn't come from the plant. They genetically engineer bacteria to make what they hope is the same protein and put that down the throats of the rats. And then, of course, the other big assumption is that it will cause disease within 14 days, and if it doesn't cause disease within 14 days, it will never cause disease. Now, sometimes they go a bit further and they will actually feed the crop itself. So now they might feed the corn kernels, the actual corn grain, or the soybeans to animals. If they do that, they tend to feed the crop that has not been sprayed, even though if you're assessing the safety of a spray of Roundup Ready soy, for example, when you eat it, it will come out of a farmer's field having been sprayed. When they do animal testing, they don't spray it first, generally speaking. Now, they use what I call unusual human health models, so they might get animals that are not physiologically comparable to humans, such as chickens, cows, or trout. And let's just check that out for a moment. So if they do a feeding study on chickens, how similar is it to you? Well, let's see, do you have wings? Do you fly? When we reproduce, do you lay an egg and sit on it? Do you have two stomachs? Do you swallow grit and small stones to help grind your food in one of your stomachs? This is the difference between chickens and you, and yet chicken studies are used by food regulators to show that it is at least as safe and wholesome as ordinary food for humans to eat. The endpoints are also not relevant to human health, so death is an endpoint, which means that if you don't die, you're healthy. So people or an organism, an animal with cancer, fine, it's not dead, it's healthy. Diabetes, heart disease, autoimmune problems, if you're not dead, you're healthy, according to that measure. Body weight, the heavier you are, the better you are. There's a measure of human health. Breast meat yield, well, that means that women must be healthier than men. Milk production, I'm sure that's very relevant to men. So these are not very good studies to actually measure human health effects, and yet they're used by regulators to show that they're safe for people to eat. These are really animal production studies. They're really designed to reassure farmers that if you feed it to your chickens, your cows, your trout, that the animals will get to a good enough size to get to market and to make you money. Now, sometimes they go a bit further. So these are long-term toxicology studies, the sort of things that should be done. I didn't see any of them really in the animals that I looked at. Nothing approaching sort of 3 months. And you really need at least 3 months, more like 6 months in my view, to do a long-term toxicology study. If you don't feed them for long enough, you don't feed them for long enough to get cancers to develop, so you can't determine if there's an effect on cancers, for example. Now, in the 28 GM crops I looked at, the longest feeding time was 1 month. The number of animals was too low. So if you've got 20 rats in a group, and you're going to, so 20 rats fed GM, 20 rats fed non-GM, and you're going to look to see if there's a difference in death rates, well, 1/4 of the rats have got to die in one group and none in the other before you reach statistical significance. If you're only going to compare boys to boys and girls to girls in those two groups, and therefore you've only got 10 rats of each gender in each group, then half of the animals have to die in one group and none in the other before you reach statistical significance. So if only 40% of the animals fed the GM diet die, I'm sorry, but that's safe to eat. You see, because it didn't reach statistical significance. Now, often in these studies, they only look at body weights and death rates. Sometimes they might weigh some organs, but that's not good enough either, because a healthy kidney can weigh as much as a diseased kidney. Often, well, there was no blood biochemistry done in any of the studies I looked at in this review. And there were some adverse findings found, however, but they were not investigated further. They were regarded as not relevant or not due to GM without any cause being given as to what they could be. Now, I'm quoting here from a paper that I've published, and that's the paper there. I'll just flick through the US requirements. We also did a review of histopathology studies. So these are where animals are fed the diet, and you look at tissues under a microscope to see what you can see under the microscope. So in this review, which we've published, and you can see that down the bottom there, we looked at the clearest evidence that you can see of whether there's any toxicity is to actually look under the microscope. We looked at rat studies because that's the standard toxicology study for this type of thing. We looked in the digestive tract because that's where you swallow your food, you digest your food, where it's in contact with you for the longest period of time. And we looked at 3 common GM genes, and that was Roundup Ready, the gene, and 2 BT genes, including any variety, single or stacked, whatever. We had a look at those. And we found that there were 47 crop varieties that had been approved for animal or human consumption that had 1 or more of those GM genes. But we found that there was no published studies for 81% of them, none of, so for 81% of these, there were no studies published at all. Only 19% of them had any studies published. And of that 19%, 3/4 of them were done after regulators had said they were safe. So in other words, regulators were making decisions based on no histopathology studies having been published. And half of the studies were published at least 9 years after approval. So if those studies had found an adverse effect when you look under a microscope, it would already have been in the food supply for 9 years. We found that not a single study was properly conducted or reported, so they weren't clear about what they did, weren't clear about the methods that they used, what they found. They often gave no results. They said, we didn't find anything of any note, but they didn't tell you what they found, so you could look at it and go whether you agreed with them or not. They said, we found no toxic effects without telling you what toxicity was for them. So, and they did see differences, but they were then dismissed as not being relevant without a good reason. So what we regard this as being is that there's a lack of evidence that they're actually safe to eat, and there was in fact not a single one of these studies we regarded as being properly conducted or reported. Now there's a new type of crop that is going to be coming into production in, I think, probably fairly reasonable amounts, and that's using double-stranded RNA technology. Now I'm not going to talk about it very much because Jack Heinemann is here and he will be giving a talk on this at some length coming up very soon. But what I wanted to say was that when you look at the regulation of these crops, and we published a paper here, that we looked at 3 government regulators in 3 countries, and we found that they didn't consider the safety of these at all. They simply assumed that they were safe and therefore didn't do a proper safety assessment on them. So now this is the study that we did. This is the feeding study. What happens when you exceed these inadequate methods? And I think you'd probably agree with me, it wouldn't be terribly hard to exceed them. So going back to GM plants that make a new protein, and this is the study I'm going to give you, where we did a long-term toxicology study on pigs fed a combined genetically modified soy and maize diet. Why pigs? Well, they're physiologically very similar to humans, much better in fact than rats or mice. And also because my colleague Howard Leger, who is in the United States and who is a farm advisor over there, said that he'd been seeing problems in pigs fed GM crops. And when you stopped the animals eating GM and put them on non-GM, the problems went away. When you put them back on GM, the problems came back. And the sort of problems he was seeing were reproductive problems. Specifically in sows, he said they were seeing a reduced ability of the sows to conceive and higher rates of miscarriage. And in boars, if boars were still used to service the sows, they were seeing a reduction in the number of piglets born because, don't forget, there's been, as GM has come into the United States in a big way in their food supply and farm animals, there's also been a change in how those animals are produced. So there's a lot of artificial insemination now being used in animals, and that guarantees a certain number of viable sperm. So if there are problems happening in farm animals in the United States with male reproductive problems, it's being glossed over or not being seen because of the use of artificial insemination. He also said we're seeing real problems in the stomach and the intestines of these animals, particularly an increase in inflammation in the stomach or small intestine and stomach ulcers, things like that. And they're seeing more of what's called hemorrhagic bowel disease, where an animal will literally go down in a screaming heap because their bowels ruptured and they'll bleed out very quickly. The pigs we used were traditional standard pigs that you use in piggeries in the United States. Got them just weaned, fed them for their entire commercial lifespan of 23 weeks. Now the males were neutered at 3 days of age so that the meat would be free of boar taint. We had a nice sample size now, not 10 or 20 in a group, but 84. Equal numbers of males and females. We individually identified each animal so we could follow each individual animal through the whole process, and we measured a lot of things daily, which I can describe later on. So here are the pigs. Here's some of them. Coming in an old school bus. I did have a giggle. So they've just been randomised now into individual pens, and then in the completing sort of growing and finishing phase, you can see how big they get, and they get to be big animals. They're in a different facility here where they can roam outside. So the feed, it was a mixed soy and corn diet. Mixed soy and corn diet, which is normal in US practices. Now, half the animals were fed 3 GM genes at once, and the others were fed non-GM. We couldn't get an isogenic control for the GM corn, but because that's not possible because of the way that it's made. And we made sure that the diets were made in exactly the same fashion. We killed them in an abattoir, weighed internal organs, examined a number of organs, and the veterinarians were blinded, so they didn't know which pigs were fed GM and which were fed non-GM. This is what it looks like in the abattoir. Cutting the kidney open in half to have a look inside. They're the lungs, cutting them open to have a look inside. And that's the small and large intestine. They were still full of digesters, so we couldn't actually look inside those. But we did find that the weight of the uterus was 25% higher in GM-fed pigs, which was highly statistically significant. Now, we couldn't have a look at what was happening with males because they were neutered. They'd had their testicles cut off quite young. We did find stomach inflammation as well. Now, before this study started, the veterinarians got lots of stomachs from pigs that went through that abattoir before our study started, and they said, look, inflammation in pigs is naturally falling into 4 categories. And here's the guy who was looking in the stomachs, and you can see he's turning over all the folds to have a really good look around, and he's going to grade ulcers in a variety of ways and the level of inflammation. But when it comes to inflammation, this was the natural order that they fell into. That's nil, that's mild, you see there's a little bit of red coming out, there's moderate, and this is severe. So you can see how red it is in the fundus over here, and that's got a lot of mucus as well. We found that there is a certain amount of inflammation that's occurring naturally in pigs in the US because of the type of diet they fed. It's very finely ground, so they get a little bit of inflammation. But what we found was that the GM took them from the low levels of inflammation and boosted them up into the severe category. So that overall, males and females put together, they're over twice as likely to get this severe stomach inflammation, and that tells you it's statistically significant. If you look at females, it's over twice as likely, but in males, it was 4 times more likely that a male fed the GM diet would get that horrible inflamed stomach, severe inflammation. We did wonder what the cause is. To us, it's most likely that the BT proteins are likely to be the cause of this because they're known to affect the gut of grubs. And it might be because we had 2 together, maybe they're acting synergistically, we don't know, but we think that that it needs to be further investigated. We did take blood biochemistry from the animals and measured it in a number of different ways. And we found that the standard biochemistry that the industry may do on some occasions did not reflect what we were seeing happening in the stomachs, because standard biochemistry doesn't pick up things like inflammation. It doesn't measure things like estrogen and progesterone, which might be affecting the uterus. So the type of standard biochemistry was simply not sufficient to be able to pick up. to be able to pick up the sort of things we were seeing in the actual organs. And in conclusion, when we eat GM crops, we eat it as a mixture. We don't just eat one crop at a time and one GM gene at a time. We eat them as a mixture. And when we fed a mixture of GM soy and GM maize, we found there was caused stomach and uterine pathologies. We have a similar digestive tract to pigs, and in my view, they should undergo long-term safety assessments by independent researchers over a long period of time before a GM crop is commercialised. This can be found, that's not actually my website, that's almost like a fan site if you like, but they do have a number of papers that my colleagues and I have published on the website, and they do have rebuttals to, my governance regulator has come out telling us we did this study all wrong, but our rebuttal to them is on there as well. Thank you very much.